Susan has a joy of science that she exudes in everything that she does. She asks really interesting questions. She's one of those people for which the success of each person around her is much more important to her than her own success.
She's one who doesn't work on easy problems. She likes to actually think hard about what it is that was really essential to a problem, and then to digest it down to what are the key questions that needed to be answered. And I think everybody would agree that Susan is one of the world's top experimentalists when it comes to studies of protein folding.
My parents are not scientists, not even close. They were in book publishing and art dealing. It wasn't until I was in high school that I realized I really liked problem solving and liked mathematics and physics. And it was a physics teacher in high school that really got me interested in just curiosity-driven questions and science in general and problem solving. Upon graduation from Cornell, I enrolled in the MD, PhD, MSTP program at Johns Hopkins. And right after graduating that summer, I went early to get some research and learn about biochemistry research. And right away, I realized I wanted to be at Stanford. And there was multiple reasons that I wanted to go to Stanford. One, and probably the most emotional reason at the time, was that my boyfriend from college was already starting at Stanford in their MSTP program. He is now my husband, so that was not a foolish decision.
Ultimately, she did decide that she would go and shift slightly from hardcore physical chemistry to biophysical chemistry and work on the protein folding problem, which was the problem that Buzz Baldwin was working on, and she actually ended up joining Buzz's group.
Buzz was an amazing mentor. He was always there for you, but he would never directly tell you what to do. When I first joined the lab, I found that frustrating. I remember, I didn't know what I wanted to work on, and I went in and said, "Can you just give me a project?" And his attitude was, "If you think of it yourself, you'll do such a better job, and I know you can think of something." And I go, "Yeah, but just please give me a project."
Well, the first thing that struck me about Susan when she joined our research group was that she was sizing it up, looking at the weaknesses, and figuring out how she would reorganize it to improve it. I started without having a research group at all. Susan looked at this and said, "This is a mistake. We need to have a research group," and she organized it.
When I joined Buzz's lab, they had made this really unique discovery, which is that a small piece of a protein was able to adopt a shape or a structure, an alpha helix. And before that time, people had thought that you needed a complete protein in order to fold, and that parts of a protein wouldn't be folded on their own.
The problem was you have a protein, you know what amino acids are there and what sequence they're in, and you know that it ends up being a final folded structure. How is it that the protein is able to do that? How does it fold up by itself? What Susan did was to actually take the problem and simplify it even further. And she got to the point where she could design a sequence, not a sequence found in nature, but a new sequence. And she could design that sequence and show that it would fold into a structure that she had predicted, namely the alpha helix.
That was a brand new thing that sort of opened the door to today's concepts of designer proteins.
Somebody had told me that UC Berkeley was going to be hiring multiple people they hadn't hired in a few years, and that I should give them my CV. I didn't actually have a CV at the time. I was at MIT 2 days, and the second day I got a phone call from somebody at Berkeley saying, "Would you come out for an interview?"
She really stood out because she could make a fairly esoteric field sound not just compelling, but exciting. We made an offer to her. She went on to do a postdoc at MIT and then came back here a couple of years later.
Protein folding is a very specialized field, but I think it's also important to realize that it is the basis of information flow that we understand the least. We know how DNA goes to RNA, and we know how RNA goes to protein sequence.
The question has always been, how does a protein know how to fold itself into that three-dimensional structure?
The misfolding of proteins, the accumulation of structure that is not what that polypeptide is supposed to do, can cause a number of well-known diseases, or at least is implicated in that. Things like Alzheimer's, mad cow disease, or Parkinson's, and many others.
The human body and the cell spends a lot of energy in the quality control to make sure the protein folds in the correct way. In order to understand why it's going wrong, we sort of need to understand how it's supposed to happen.
That could give us insights into potential therapeutics that would allow us to mitigate some of these diseases potentially.
From the days that I joined Buzz's lab until today, I've always been working on some aspect of the protein folding problem. I really wanted to look at a single molecule as it folded. I wanted to know if the protein did the same thing every time it folded, and when it populated intermediate, did it have to?
Traditionally, biochemistry and molecular biology is driven by procedures in which we study the average behavior of, let's say, 1 million or 1 billion different molecules. You learn a lot when you start looking at one molecule at a time, and that's a big frontier that Susan has been a very active part of.
One of the things that drew me to the lab was this kind of infectious enthusiasm for tackling those challenging problems head on, even though the chances of success in some cases might be lower than something that was a little more predictable in terms of the science that you might be doing.
So I had a colleague here, Carlos Bustamante, who had also developed a lot of experiments using the optical trap. A plastic bead is held fixed in the laser of the optical trap, and then in our case, we could have another plastic bead held on a tip of a pipette, and we could string a molecule between that.
We eventually got to the point where we had developed a method that would allow us to very specifically tether these individual proteins using kind of what we call DNA handles to give them space between the beads.
We could hold the protein in place attached to 2 plastic beads, and we could watch the protein, what happens if we pulled on the beads and applied force. And so that was done with a graduate student from my lab, Beth Shank, and a graduate student from Carlos Bustamante's lab, Ciro Ciccone. And they worked together for 5 years to get this to work.
There was a moment where I was holding the protein at a single force. I'd pulled it a little bit and then was just holding it stationary. And what I could see was that there was structure based on the length that we were detecting between our 2 beads. You could tell that something was forming and then unfolding and then forming and then unfolding. I knew at that point that what I was watching was basically this partially folded structure of a single protein that was being tethered between these 2 giant beads, actually folding and unfolding right in front of me. And that was a really exciting moment. Yeah.
And so it was very exciting for me because this protein I had studied for a long time, all of a sudden I could physically watch it go through this conformational transition, 1 molecule at a time. It was a movie, and we could watch that movie over and over again because it was a detail that I think nobody had ever seen before. And so it was very empowering. What was also really exciting about it is it was a technology I didn't know anything about. So I was learning. My students were teaching me as fast as I could learn how to do this technology.
What this did was to bring the protein folding problem, the experimental studies of protein folding, down to the level where you are now looking at it on the level of a single molecule.
It would allow us to look at these very early folding processes that really had never before been able to be observed directly. The main thing that she did that was so critical was she brought together people that could interact both intellectually and socially and creatively, and then gave us the space to actually do a lot of the hard work ourselves, while still being there to provide the feedback and to provide the guidance, but not to be a micromanager at all. I think you need to have a space that intellectually people feel free to do something crazy or ask a question that's maybe out of the norm, or to try and do something that no one thinks is possible.
Mentoring students is as rewarding to me as the scientific discoveries themselves. It's very exciting to see a student, help them figure out what their skill set is and help optimize their skill set to move on to do what's best for them, whether it's academia, teaching, industry, policy, whatever.
You know, there's a lot of things that I say in my lab now, and I'll hear myself giving advice to my students or my postdocs, whether it's about their science or it's about their presentation or it's about how they put the poster together, and I basically feel like I'm channeling Susan. So I literally am saying things that she said directly to me, and now I get to pass them on to my students.
So here at Berkeley, I serve as the director of an institute called QB3, which is the Institute for Quantitative Biosciences. So we spent many years thinking about how do you build a building that promotes interactions? How do you build a building where you can have people working on lots of different fields together, and the sum is more than the sum of the parts? And in designing it, I actually modeled a lot of what I was thinking about of my experiences when I was at Stanford in the biochemistry department, because on a much smaller scale, that's the way the biochemistry department worked. People from different labs mingling, and from those interactions, new science comes.
Susan recognized the transforming impact that high-quality cores would be, and it was one of her major goals in establishing the QB3 facility. So now we have the capacity for individual graduate students, for example, to go to our core facility and learn how to devise microfluidic devices in ways that we wouldn't have been able to do before.
She's not only a terrific scientist, she's also a terrific mom. She has 2 wonderful children. She's a very, very compassionate person, a great person to have as a friend, and someone who has leadership abilities and qualities that allow her to do more than just science. And, taken together, it's just a remarkable achievement.
I think she's kind of fearless and wanting to address these really big picture questions that have been challenging the field for a long time.
She has a taste for what's an important problem. She has a sense for what's a creative approach to it. And she's not afraid to ask what seem like simple, fundamental questions. Because only when you try to answer those simple, fundamental questions do you realize what you do know and what you perhaps don't know as much as you thought you did.